Okay, this will be our last video summary for this unit as we're getting near the end of things. The other day we looked at the cell membrane specifically and what makes up the cell membrane, and we want to talk about today more deeply how things are regulated moving into and out of the cell through this lesson on cell transport. Now, if we compare cell transport to the movement of things across a membrane, you can use this without getting too political to look at the movement of immigration across different countries. And I kind of put a couple videos in there, some extremes of Donald Trump looking to build a wall versus the European Union where you can just go anywhere. And the reality is with the cell membrane, none of those options works well. We can't have a rigid cell wall where things can't get in and out of, but yet we need control. And so both have their pros and cons, but we need a cell membrane that is selectively permeable. Impermeable would mean it's a wall, nothing gets through. Permeable means everything gets through. And so we want to be selectively permeable, and we're going to look at different ways that we can be selectively permeable with our cell membrane. For whatever reason, sometimes students miss out in this lesson that things have to go both ways, and we fixate on things moving into the cell. But things have to be able to move into the cell and out of the cell in order to control homeostasis. Again, this is really just homeostasis, maintaining the environment for our cell. We'll look at in our last unit how we maintain homeostasis for our entire body. But there's two ways that we regulate transport, and there's passive transport and active transport. So we'll look at the characteristics of both and understand that both of them have their own pros and cons. When we look at passive energy or passive transport, passive by nature doesn't revolve a lot of energy. It's kind of random, moves wherever it wants to go. It kind of moves 360 degrees, moves randomly, but it moves from high concentration to low concentration. And so there's lots of examples you can use to think about passive transport, whether it's you're steeping your tea, whether it's an air freshener, whatever it might be. Biologically, there's three different types of passive transport. There's diffusion, there's osmosis, and facilitated diffusion. It's worth mentioning that osmosis is really facilitated diffusion, but it's specifically using water. And so water is so important, it's the medium of life, and so we need to look at specifically how water is regulated. I'm not so concerned that you can understand there's different terms within regulating water levels that you'll see in the note. But I want you to be able to make sure water always moves from a high concentration to low concentration. And there's specific proteins that help it do that, aquaporins. And again, we see the role of proteins in this. So three types of passive transport: diffusion, osmosis, and facilitated diffusion. When you get to this part of the slideshow, I'd like you to stop for a little moment. I want you to kind of brainstorm and think about it, maybe just at your table groups, and think about why could life not just exist with passive transport alone? What would the pros and cons of that be? Each of these transports has pros and cons. So why could life not exist with transport alone? Make sure to spend some time and talk about this. So now we introduce active transport, and active transport has two real characteristics that are important. It moves specifically and it moves against the gradient. Those are big deals. Now, it also requires energy, and so we see ATP energy is required to do this. So you'll look at that and what are the pros and cons of active transport here? Like so many things, there's different types of passive transport. We have different types of active transport. One of those is protein pumps. And here again, you see the chemistry driving the biology. We have specific proteins that will move things from one side of the membrane to the other based on their chemical nature and the structure that they have. One example of that is a sodium-potassium pump. In our homeostasis unit, we'll look more in depth at the sodium-potassium pump and then how that allows every nerve cell in the body to send messages. It's absolutely crucial for life. Notice how when you look at the sodium-potassium pump, notice how the protein shape actually changes. The chemistry changes with energy so that it will attract either sodium or potassium. So it's a great example there of how chemistry drives the biology and allows us to control the environment of the cell. The sodium-potassium pump is just one example of protein pumps that are located within the cell membrane. I moved to the bottom of the screen for this one, but for the second type of active transport, we have what's called endocytosis. Endocytosis is used to move large, bulky materials into the cell, or any kind of larger material. And again, there's three types. I didn't make up the word, so don't get mad at me, don't get mad at Mr. Allen, but there's three types of endocytosis. There's pinocytosis, phagocytosis, and receptor-assisted endocytosis. So make sure you can distinguish between those. In terms of our test, when we come up to our test, this section of the test, there's going to be more multiple choice. And so just making sure you can recognize those terms I'm not going to require that you spell them all exactly, but you should be able to recognize those and identify what's pinocytosis, phagocytosis, or receptor assisted. Our final type of transport is, again, kind of like endocytosis, but instead of moving large things into the cell, it's exocytosis. We're exiting the cell. And so exocytosis is used to move large molecules out of the cell. This could be hormones, waste, different proteins, whatever it might be that we need, different enzymes that are being produced in the digestive system, but we need to get things out of the cell. And so that involves exocytosis. And you can see it again in the animations, how the exocytosis, we have part of the cell membrane, really, the phospholipid, forms these vesicles and it releases its contents out into the environment. All right, as you finish this lesson, I want you to take some time as a class. You can talk about this, but consider what you know about passive transport. Why, again, can life not exist with just passive transport alone? And then also add on to it, why could life not exist with just active transport? We have both of these, and they're both critical for maintaining the life of a cell and maintaining homeostasis. Make sure you could provide at least 3 reasons why life cannot just exist with passive transport or active transport. So you can consider that. We're done this section of our note. Now you're going to start reviewing and getting ready for our test, but I hope these video summaries have been helpful.